Okay, we're going to continue discussions with the next topic, which is normalization. So, an endless topic in all of genomics, how to normalize data appropriately across data sets and samples and experiments and so on to make things comparable appropriately. Obviously, we'll be focusing on what's necessary and available for single-cell data. So, Tallulah took us through the identification of important confounding factors and explanatory variables, and SCADA in a couple of ways allows one to account for these variables in statistical models or condition them out using this function normalize express, if so desired. And what we can do if we have a series or a set of variables that we want to regress out of our expression values, then we can provide what's called a design matrix. So that's just, well, if you don't know what that is, then I can explain that down the line, but fundamentally it's turning those individual factors that we want to regress out into a matrix that contains all of that information that we can provide to a linear model. So if that's gibberish to you, then we can discuss it offline, but I won't go into the details now. But that's quite easy to construct that sort of matrix in R and provide that as an argument to this normalize express function. So actually it's not too onerous to do that if that's something we need to do. And there are examples in, I guess, in the SCADA documentation. You can look up the documentation for this normalize express function and see how you would construct such a design matrix and how you would use that function to regress out factors that you've decided are noise or things you want to remove from downstream analyses. What we'll focus on here is the use of simple, what are known as size factor normalization approaches to correct for library size, and how this can improve or remove the effects of some of the confounders that might be problematic in analyses, can reduce the impact of some explanatory variables where it's useful, and I guess we'll also discuss some of the limitations of these approaches per se. So as you already will know, what we call the library size, or the total number of reads or counts that we can assign to a particular cell. These library sizes vary a lot because single-cell RNA-seq data is often sequenced on highly multiplexed platforms, and so that means that, so just by chance, just with stochastic nature of sampling and library prep, the total reads which are derived from each cell may differ substantially. And so that's even without the fact of very different qualities between the cells. Even if all the cells were sort of starting with the same amount of RNA that we're able to extract, even so, throughout the sequencing, library prep and sequencing process, we're going to end up with substantially different library sizes from the individual cells. And so some quantification methods like cufflinks and RACM incorporate library size and build this in when determining gene expression estimates, and so don't necessarily require this sort of normalization. And that's some of the idea behind a TPM quantification method. But if another quantification method was used, or especially when we're operating in the count space, then library size is a necessary thing to correct for by either multiplying or dividing each column of the expression matrix by a quote-unquote normalization factor. And this can be understood as some sort of an estimate of the library size, or the effective library size relative to the other cells. And many methods to correct for library size have been developed for bulk RNA-seq, and with some caveats, the similar ideas and even some of the same methods can be applied to single-cell RNA-seq data. So we can talk about upper quartile, size factor approaches, counts per million, RPKM, FPKM, TPM, transcripts per million. All of these are various approaches to trying to get values, expression values from different cells on the same scale to make them comparable to each other. So CPM is sort of one of the easiest ways to normalize data. in this way. So we're going to take the counts and convert them to counts per million. So that's as simple as it sounds. We would just do that by dividing each column, so the values for each cell, by the total counts and multiplying by a million. That gives us counts per million. If we do this in single cell expression data with spike-ins, then we should exclude the spike-ins from this calculation of total expression. Like we want to be looking at the total expression for the endogenous genes for a cell in order to correct for the total cell RNA content. And so the spike-in counts would affect that adversely, so we'd want to exclude that. And using like the calculate CPM function in Skater takes that into account. But if we wanted to do this from scratch, then a calc CPM function like this does it. So you can see in this case, we define normalization factors as the column sums of our expression matrix, having removed any genes that we've identified as spikes. And then this line applies this approach of dividing the counts for each particular cell by the appropriate normalization factor or column sum and returning that normalized matrix. So that's straightforward and useful, and it's applicable to any count data, so it's very general, which is useful. A potential drawback is that if our sample contains genes that are both very highly expressed and differentially expressed across cells, then in this case the total molecules in a given cell may depend on whether these particular genes are on or off in the cell, and normalizing by the total counts, the total molecules, we may be hiding the differential expression of these genes. And what can be even worse is that if we fail to account for this possibility of differential expression between different sets of cells, we might, through the normalization process, falsely create the impression of differential expression by scaling these values in the wrong sort of way. And so that's the major drawback about sort of raw CPM, which some of the normalization methods that we'll discuss try to take into account. So you'll also see RPKM, FPKM. So RPKM stands for reads per kilobase per million mapped, and it's my least favorite expression unit of all time. FPKM accounts for paired end reads, so the F stands for fragments instead of reads in RPKM. TPM stands for transcripts per million, and RPKM, FPKM, and TPM all try to adjust the counts by the length of the respective gene or transcript. So to build in this idea that if you have a longer gene or a longer transcript and you're sampling reads from the full length of the transcript, then you would expect to see more counts coming from longer genes or longer transcripts. And so that makes it very difficult inherently to try and evaluate sort of or compare expression values between genes that have very different lengths. Because even if they're, quote unquote, if the number of transcripts in the actual cell were equal for a long gene and a short gene, after sequencing you would expect to see more counts for the longer gene than the shorter gene. And that's challenging inherently. So those methods try to take that into account. I guess it's as good a point as any to say that RNA-seq, whether bulk or single cell, is a relative measure. Like we can only assess, really look at how highly or lowly expressed things are in a given sample or given experiment. And it's always been a challenge to compare expression levels of different genes because there are so many inherent differences in them. So that comes down to gene length. GC content can affect how well we can assay and measure the gene, the expression coming from such genes. So all of these normalization methods sort of address this or don't in various ways, but it sort of remains a difficult thing. So another approach we call RLE here, which stands for relative log expression, and this is the size factor that was proposed and popularized in the DE-seq paper in 2010, so coming out of bulk RNA-seq methods. And this was a method to try and address that issue that if we have a set of genes that's very highly expressed in some cells and not expressed in others, then normalizing on total counts per cell can induce the appearance of differential expression. And so the approach they took to combat this was we first take the geometric mean of each gene across all cells, so it's a sort of robust measure of the average expression per gene. And then the size factor for each cell is so for a given cell, the median across genes for that cell, the median of the ratio of expression relative to the gene's geometric mean. And so that has the effect of ignoring outlier expression, and so we're safer compared with normalizing to the total count per gene. We're a bit more robust to that sort of effect of a set of genes that are very highly expressed and differentially expressed between cells. A drawback is that since it uses the geometric mean, only genes with non-zero expression values across cells can be used in its calculation, and so that really makes it unadvisable for large, low-depth, single-cell RNA-seq experiments. And so in EDGAR and SCADA, which uses the method in EDGAR, this method's known as RLE, or relative log expression. There's an example function here of how you could compute that from scratch yourself. But in practice, this is difficult to use for single-cell RNA-seq because in many cases we have a large number of zeros, and this method coming from bulk RNA-seq is not well suited to that sort of data. A relatively straightforward approach is UQ, the upper quartile method, which also was proposed in 2010 by Bullard et al. And in this case, each column, so each cell, is divided by the 75% quantile of the counts for that cell. And so instead of using the total, we're using the 75% quantile, so that ignores the really highly expressed genes for each given cell when computing that overall effective library size. We can scale that quantile by the median across cells if we want to keep that absolute level of expression relatively consistent, which can be useful. A major drawback of this method is that for low-depth single-cell RNA-seq experiments, a large number of undetected genes may result in this 75% quantile being zero. So imagine if you start with a full set of 30,000 genes, then for a given cell, it's not uncommon for for the 75% quantile to be zero. And if that's the case, then dividing by that value starts to make very little sense. Of course, you could overcome that to some extent by generalizing the idea and using a higher quantile, like 99%, which is the default applied in SCADA, or by excluding zeros prior to calculating the 75% quantile. But again, this is a case where just porting across the ideas that were successful in bulk RNA-seq don't necessarily work as well in single-cell RNA-seq. And here's an example of how you could write an upper quartile size factor function from scratch to do that normalization. And so here we're applying the 75th percentile on non-zero values in the matrix. But you could obviously play around with that value. So a final approach coming out of bulk RNA-seq is called TMM, which stands for the weighted trimmed mean of M values, which I'll explain in a second. And so this also came out in 2010, so from Robinson and Oshlag. And the M values in this case are the gene-wise log2 fold changes between individual cells. So we take a pair of cells, and compare the expression values across genes, and that ratio we can define of value in one cell versus cell 1 versus cell 2, we can define as a fold change, and putting that on the log2 scale gives these M values. And the idea is that in appropriately normalized data, most of the genes, or like on average, the genes should not show any differential expression between cells. So this is making the assumption that most genes should not be differentially expressed between cells, which is a common assumption throughout many approaches in RNA-seq. You might think about settings where that assumption might not be appropriate, especially in single-cell RNA-seq data. So computing these M values, we use one cell as the reference, then M values for every other cell can be compared to this reference. We trim the outliers by removing the top and bottom 30%, approximately 30% of cells. That can be the values that can be tweaked. And then the average of the remaining values is calculated by weighting them to account for the effect of the log scale on the variance, given that we know that on the raw scale of counts, the variance will increase with the mean. Once we've done that, each non-reference cell is multiplied by its calculated factor to give us another size factor normalization approach that we can apply per cell to divide by. As with other methods coming from bulk, we have the potential issue that if there are insufficient non-zero genes left after trimming, then we might not get sensible results, and we make that assumption that most genes are not differentially expressed. And so in the case of bulk samples, that's a pretty safe assumption to make. You could imagine situations where if we had very distinct cell types with very distinct gene expression repertoires, then maybe that assumption is not so safe in certain single cell expression settings. So a nice extension to these ideas that came from bulk is this one, another size factor normalization approach implemented in the SCRAN package. And this really just applies the same idea that we want to define a size factor per cell, which we can divide our expression matrix by these size factors to get more comparable values between cells, but use an algorithm to define these size factors that's more appropriate for single cell data and the particular attributes that single cell data has. And so that's what Aaron Lund with Carsten Bach and John Marioni did last year. And just to try and describe it briefly, this method deals explicitly with that problem of large numbers of zero values in single cell data by progressively pooling cells together and aggregating the counts from pools of cells and calculating a normalization factor for that pool. And then by progressively pooling with different cells in different pools, they build up a system of linear equations that can be solved to give a size factor for each individual cell. So that's glossing over a lot of details, but the gist of it is that we can get a size factor for each individual cell that's computed in a way that is robust to the large number of zeros present in any given cell. And that works because by pooling and repooling the cells, each cell is found ultimately in many different pools, and we can aggregate that information to solve that system of equations for individual cell-specific factors using the power of linear algebra, which is nice. So, so my general thought is that as a whole, there are certain limitations to size factor normalization. If we're going to do size factor normalization for single cell data, then to me it makes most sense to use this method in SCRAN, which was explicitly developed to solve some of the drawbacks of applying bulk methods to single cell data. So that's that. Yep.
With large number of zero values, wouldn't you remove it and just work with what is left to the data?
So the question is, with a large number of zeros, wouldn't you remove them? before doing anything. You can do that to a certain extent, and so we do that with filtering genes with very little information, and that makes sense. But even so, the matrix can still be quite sparse even after removing genes that are really not expressed or expressed only at very low levels across all cells. In particular, in a case where there are substantially different cell types, if a particular gene is expressed moderately or highly in a particular cell type, which makes up 10% of your cells, then by definition that's going to be mostly zero across all the cells you've assayed. But you're not going to want to drop that gene from your analysis because it has important biological meaning. And so that's the sort of one way that we can get to very sparse matrices or expression matrices in single cell data, even after removing sort of obvious candidates like the dodgy cells and genes that have no information. So that's why these challenges remain even after QC and that sort of thing. So a completely different approach to normalization is downsampling. And the idea is, again, to account for differences in sequencing depth between cells, but instead of dividing the values by some size factor, we can instead downsample the expression matrix so that each cell has approximately the same number of total molecules. The benefit of this is that zero values will be naturally introduced by the downsampling, so we won't have the same effect of biases due to differing numbers of detected genes in the cells. A major drawback, however, is that the process is not deterministic because we have to use some sort of sampling approach. So we could make it reproducible by setting a seed or something, but otherwise, each time the downsampling is run, the resulting expression matrix is going to be slightly different. It'll have the same statistical properties, but it will be slightly different in the details. So we might need to run this multiple times with multiple downsamplings to ensure that conclusions we draw from the downsampling results are robust. The other objection is that you're potentially throwing away a lot of data. So if you're downsampling to the library depth, you essentially have to downsample to the library depth of your lowest depth cell. So depending on how strict you've been with your QC, you could be throwing out a very large proportion of your sequencing reads, and part of me objects on fundamental principles about doing that. In some approaches, it can be very useful. I think can be probably more applicable to UMI count data than read count data. I've been speaking to Aaron Lund recently about using this as a sort of last resort for batch effect correction. So in cases like we've got, say, UMI count data and batches that otherwise sort of resistant to merging, then downsampling can be effective approach at that point. But I would certainly tend to use it as a last resort rather than a first choice thing. Again, here is a function that you can see how you could write a method for doing this from scratch and how you could tweak that yourself. So we're going to now look into the effectiveness of these various normalization methods. And so, in general, this can be a little tricky. Comparing methods in general in bioinformatics can be tricky because often we're lacking the perfect ground truth data set to compare to. So in this case, we're going to use two particular things to look at. We'll produce PCA plots with different normalization strategies and visually inspect them to see what features arise or disappear with various normalization approaches. And we'll also look at plots after calculating the cell-wise relative log expression, which has a function called plot RLE in Skater. And this looks at sort of cells. We can see cells with many reads having higher or lower expression than the median expression for most genes And so if many genes have higher expression, higher than median expression for most genes, then we see a positive value in the RLE, and otherwise, inverse case, we see a lower, if it's lower, we'll see a negative value for the RLE. What we'd like to see in well-normalized data is sort of an RLE plot that's close to 0. So I think that'll be obvious when we look at the plots. This here is an example of how you could calculate RLE, and essentially a more sophisticated version of that function is used in the plot RLE function in SCADA. So as I said previously, the RLE, TMM, and UQ size factor methods were developed for bulk RNA-seq data, and so for sort of deeply sequenced single-cell RNA-seq data, they may well work well, but won't necessarily work well in all situations, as their underlying assumptions may be violated in problematic ways. So they're available in SCADA to use and to explore data through the calc norm factors function. SCADA wraps around this calc norm factors function from EDGAR, which implements these methods, and so they're available for use from within SCADA. There are some slight differences between that. Yeah, I don't think that's particularly interesting for general use, but if you're worried about the technical differences between implementations in EDGAR, SCADA, and other published work, then we can always chat about that. Yeah, so just for here, we use the calculate CPM function in SCADA to calculate CPM. RLE, UQ, TMM can be accessed using the normalize express function, and for the SCRAN method, we get that from the SCRAN package, and specifically a function called compute sum factors. And speaking about the interoperability of different bioconductor packages, the nice thing here is that the SCRAN package also uses a single cell experiment class as its data object, so we can have our single cell experiment object and use methods from SCRAN and then methods from SCADA and go back and forth, and that works quite nicely. SCRAN and SCADA are particularly closely integrated, and so if we compute the size factors using the SCRAN method, they're stored in the single cell experiment object, then we can apply SCADA's normalize function to do the normalization, and so that for the user becomes quite convenient. And for downsampling, we can do that function we'll use. So we'll continue working with the tongue data that we've used previously. So these few lines of codes load these libraries and load the data and subset it to what we want to use, what we've defined previously from the QC, defining our endogenous genes. So if we do look at the PCA before any normalization, so this is looking at the raw log counts, coloring by batch, then we can see that there's the cells from one particular individual are quite distinct from the other two individuals, but otherwise we're not getting so much biological structure identified when doing PCA on those raw log counts as we've defined them previously. So if instead we replace that log counts, or we just add log counts to our data set as log2 CPMs, so we've used that calculate CPM function and add an offset of 1, then we can see that accounting for library size is already bringing out more structure in the data. So now we have 3 distinct groups of cells which correspond to the 3 individuals. For 2 of the individuals, we really see a strong effect of batch within the individual. So in this case, for this individual, the 3 different batches really don't overlap much at all, although they are distinct from cells from the other 2 individuals. For this individual down in the bottom left, same story. We removed one of those batches in the QC. The 2 remaining batches really don't overlap, but they are distinct from the other individuals. And in the top left of that PCA, we see the third individual where there's actually some overlapping, so less distinction between the specific batches there. So in that respect, you can see that even just the simple thing of going to the log counts per million scale for our expression values, we immediately pull out more biological signal in that visualization than we do operating on the raw counts. So that's useful, but we can do better. If we look at the RLE plot here, so now you can see that comparing the CPM normalized values at the top to the raw values down the bottom, we see a lot more consistency across the various cells from the different individuals and batches after applying CPM normalization compared to the raw, which is really much more all over the place. So just to explain this plot a little bit, so what we have is a box plot for each cell showing those RLE values for each cell. So effectively each line that you can see is a little box plot. The circles indicate the median, so what would normally be the middle bar in a box plot is indicated by a circle here, and the line indicates the interquartile range, so what normally forms the box in a box plot. So in a usual box plot, you then have whiskers showing off, and they're not shown just because otherwise the scale of these gets crazy. So we're just looking at the interquartile range, so the box from a normal box plot, and the circles show the median value. Yeah, so the point to take away here is we want to see nice flat RLE plots that shows effective normalization, and I think the distinction here with the unnormalized raw values is really obvious here. So that's just using sort of vanilla counts per million. We can also look at RLE size factors. So we can apply the normalized express function in this way, specify the method that we want to use as RLE just on endogenous genes. And if we do plot PCA on using those values instead, not a substantially different picture from calculate CPM. So in this case, the effect of sort of trying to tweak the library sizes in a sense by using that size factor doesn't have a major effect on the normalization here. If we do look at an RLE plot again, comparing the raw on the top to the RLE normalization method here, we can see the benefit of RLE relative to the raw value. If you were to compare that back to the vanilla CPM, not a profound difference in normalization. We can do the same thing with upper quartile. So again, applying the normalized express function and setting method to upper quartile. If we do PCA here, this looks a little bit different. So now you can see there's a bit less separation between the cells from the separate individuals. So the PC2 has sort of more or less been flipped. So we used to have the triangles up in the top left and the circles in the bottom left, and they've swapped around. Now there's a bit more distance between the two batches for that individual NA19098. So this is probably not looking as good, although this is where it gets a bit hand wavy, but arguably not looking as good as the previous methods, since now the distance between the individuals, which we expect to be the major driver of differences between cells, is less than previously. Yep.
Was there not much variance explained by the principal components? I know that with some other data sets as well, percent component.
Yeah, I think they're inherently variable data sets. So it's a common observation that before QC, what tends to drive variation is that the total number of genes expressed per cell, the total counts. So once we sort of remove that as a major source of variation, then any sort of given biological factor typically doesn't seem to explain a large proportion of variation. So I don't think I have a deeper explanation than that, except that it's certainly a common observation, and I think reflects probably just the inherent variation in these data.
Because it was
Likely, but I guess not necessarily only so. Yeah. So how do we compare this PCA plot to, so the first two, as we saw, were very, very similar. So the first principal component looks to be capturing the same source of variability. It separates what individuals that, like individual 19239 is on the right-hand side of principal component 1, and the other two individuals are on the left-hand side. So we see that still present with the upper quartile normalization. The difference is in component 2, really. So here we have the triangles in the top left and the circles in the bottom left, and that's sort of flipped around in the upper quartile plot. That, in principle, like flipping a principal component in principle is not in itself notable. It just happens to be a difference that we observe in this case. But the only difference, major difference I really see in this plot is that there's a bit of a less distinction. I would say like these two clusters of points for this individual are now further apart than they were previously and closer to this individual. But I mean, we're talking about like very subtle differences here that I don't think don't want to over-interpret these sorts of things. So overall, I'd say this gives a very similar picture to the CPM and RLE methods that we've previously seen. If we were trying to be really nitpicky, I'd say this looks slightly worse, but you know, we're getting very much a similar picture. Yeah. Yeah, so the question is, would we ever realistically expect to see all of the replicates for a given individual overlapping? And so like here, it's quite clear that yes, for that individual is separated from the other individuals, but there's distinct batches there. Look, like the ideal sort of pre-processing or sequencing or batch correction method would give us no distinction between batches. So as a cynic, I would expect to see batch differences because I always expect to see batch differences. But it's also, I think, a reasonable goal of pre-processing to try and minimize those batch differences through principled approaches to normalizing and correcting the data. So certainly at a first look at these data, yes, I would always expect to see batch differences, but I would also hope through careful pre-processing that we could minimize that effect and remove those unwanted sources of variation and focus on the biological sources of variation that we're really interested in. And so, yeah, so it's our aim. So again, compared to the raw approach, UQ does give a much happier looking RLE plot. So TMM and the final bulk RNA-seq size factor method can also be implemented or applied using this normalized express function and setting the method to TMM, much as in the same way as previously. Again, we get overall a similar looking plot to the other size factor normalization approaches. That first principal component still separates individual 19239 from the others. Yeah, I think we've said what needs to be said there. The picture is not profoundly different. Compared to the raw, again, TMM much better. You might sort of inspect the TMM compared to the RLE plots for others, which I'll let you do during the exercise section. You can see how you can apply different things. So here we've made the comparisons back to the raw, but you could do it to specific normalization methods to compare. So the SCRAN approach does a quick clustering of cells using this function called quick cluster. So it's a SCRAN function, and we use that loose or rough clustering of cells as inputs to the compute sum factors function. So we apply compute sum factors with that clusters argument that returns, we get our single cell experiment object back with the size factors from Scran, computed by Scran, added to it. Once we've got those size factors stored in our object, then we can apply the normalize function in Skater to get our normalized values in the log count slot. And so if we then do PCA again on that, we get a qualitatively similar picture. We've got sort of 3 very distinct batches still for that particular individual on the right-hand side. We're getting a bit more distinction perhaps between the triangles and the circles here. The different batches for the triangles there are starting to overlap a bit more, which is what we'd like to see. Those 2 in the bottom left corner still distinct batches, but a bit closer together than some of the other methods perhaps. So again, not profoundly different story, big improvement over not doing any normalization. In this sort of relatively deep data, all of these approaches perform relatively similarly. For me, my default preference is to use the Scran method, but you could also justify or defend using other approaches, particularly in a case like this. To look finally at the downsampling approach, so we can use that downsample matrix function defined above to generate new log count values and applying them here. We may be, so even less variance explained by the first principal component than previously. The first principal component does still differentiate really between that individual 19239 and the other 2. Those batches for individual 19239 are starting to overlap a bit more than previously, so that might be useful. On the left-hand side of the plot, we're not getting such distinction between the circles and the triangles anymore, and you can see that this downsampling approach has led to some individual cells, or we can see more visibility perhaps for those individual cells that started off with fewer total features. So that's one other thing that pops out to me looking at this plot. If we look at the RLE plot, then we are getting a large degree of consistency between the different cells. What might be in some sense surprising though is that perhaps if you can look at this blue individual here or blue batch here. Now this actually has a lot lower sort of levels of variability, which is actually less consistent with some of the other batches across here, whereas the library size only methods tended to give much more consistency across the different various batches. So assessing these, as we said, is a subtle thing, and we've just looked at this RLE and PCA. You can see that compared to no normalization, all of these methods are much more obviously in a PCA plot show the underlying biological variation that we know to be there. I think in this particular case, all of these could be defensible approaches. They all yield fairly sensible results. In extreme cases where the assumptions underlying those methods that came out of bulk RNA-seq break down, then those particular cases are where that Scran approach might be particularly useful. So all of those size factor normalization methods discussed there effectively ignore gene or transcript length, but some other methods do combine library size and fragment or gene length normalization. So these include RPKM, reads per kilobase per million mapped, designed for single-end sequencing, FPKM, so fragments per kilobase per million mapped, which is the same as RPKM but for paired end sequencing, and make sure that the paired ends mapped to the same fragment are not counted twice. And the more common these days, because it's an output of quantification tools like Callisto, Salmon, Sailfish, RSEM, and others, is this idea transcripts, just transcripts per million, which tries to identify like the number of transcripts expressed and take that into account, or take the effective transcript length into account in the quantification. So in this particular data set, these are not applicable, since the end of the transcript which contains the UMI was preferentially sequenced, so we don't have reads coming from the full length of the transcripts. And these are also things we generally don't want to do sort of ourselves, as it were. These values should be calculated using appropriate quantification software from either from raw FASTQs or from aligned BAM files. We generally don't want to do them post hoc from read counts, since often we have problems in that only a portion of the entire gene or transcript is sequenced, not the entire length. So if we just take the annotated gene length or transcript length from some database, then that may not reflect the actual sort of what we can actually sequence from a given transcript, and so that may send us off in the wrong direction. And one sensible thing to do, and there is a function in Skater for doing this as well, is you can check for the relationship between gene or transcript length and expression level, which can be sensible. So in Skater, there's this function called get BM feature annos, which we can use to get annotations from Biomart to help us get information about genes like transcript length or gene length and so on. And if we were feeling hardy, then we could apply that to compute some of these other values. I don't particularly want to discuss that in detail now, so if you have questions about that, then happy to discuss that, particularly if it's relevant to things you're working on. But I think we can skip over that. So that brings us to time for an exercise. So over to you to perform that same sort of analysis, investigating different normalization methods with the read counts of the tongue data instead of the UMI data. So thinking about this particular warning that I glossed over back up here for some of these approaches using normalize express. So for example, if we apply TMM normalization with the normalize express function, we see this warning saying spike in transcripts in ERCC should have their own size factors. So that's there to warn us that these bulk RNA-seq methods, so RLE, UQ, TMM, were never designed with spike-ins in mind, and so the default behavior is that we ignore them. So we use endogenous features to define the size factors for all cells, and these size factors are then applied to those endogenous genes. But the more principled approach should be to define, if we want to also normalize size factors, spike in transcripts appropriately, then they need separate size factors. And so that's not accounted for by any of these methods that came from bulk, and so that's what that warning reflects. Scran, however, so the way to deal with this in a better way is to use the Scran approach. So there's the compute sum factors that operates on the endogenous genes. There is another function which I think is called compute spike factors, which specifically computes size factors for the normalization of the spike-in genes. And so if you care about the spike-in genes and normalizing them appropriately, then you would also run the compute spike factors. And if you do that, if you do both of those steps, then the normalize function will, in that one step, will apply the appropriate size factor normalizations to the appropriate genes and cells. So that's the way to tackle that problem if it's a concern.